Hello children. Now on chapter 5 of our NCERT textbook, Principles of Inheritance and Variation, under that particular topic, the subtopics again, that is Testcross and chromosomal theory of inheritance, are the ones which I will be talking about today. So here, initially let us give a little introduction on what is Testcross. Now if suppose you have a genotype, okay, you have a particular genotype which can be either a homozygous dominant, heterozygous dominant, or a homozygous recessive. That is the genotype. But when it comes to phenotype, you will see only two. This I am talking in case of only two alleles. So there are only two phenotypes. Either the dominant or the recessive is seen. Now the question here is, in case of recessive, okay, we know the genotype will be homozygous recessive. But what about the dominant phenotype? It can be either a homozygous dominant genotype or a heterozygous dominant. So how do we know? How do we find out what is the exact genotype for a dominant trait? To know it, to identify that, to detect that, we carry out a particular type of test which is known as test cross. So let us now see how it happens. Test cross. So what is this? It is a method. It was also devised by Mendel, Gregor Johann Mendel, to determine the genotype of an organism. Genotype in the sense, here when we say determine the genotype means whether it is homozygous or heterozygous. And heterozygous will never give us a recessive trait, that we know very well. So a heterozygous individual will also be expressing the dominant trait. Homozygous dominant, obviously it will be the dominant trait. So to find out the genotype of an organism, it is a method which is known as test cross. Now how it is done? Here the unknown dominant genotype or the phenotype, whatever, okay, the unknown individual, the individual with the unknown genotype is crossed with a recessive parent. We take a recessive trait of the same type of individual within the same population. We will take a recessive individual. We will take the unknown dominant expressing individual, and then we will cross both of these. For example, we will take the flower color of pea plants, that is violet and white. Flower color, the two contrasting traits are violet and white. So let us take them. So let us see how it happens. Here you have, as I've shown here, okay, this is the violet flower. Okay, this is the individual or plant which is expressing violet colored flowers. Now this violet color of flowers may be due to a homozygous dominant gene WW, or it may be due to a dominant gene, that is capital W small w. Now there are two cases, case 1, case 2. So here you can see here the symbols that are used are W. Okay, so here what we do is this is the first type of cross, assuming that the violet coloration is due to homozygous dominant genes. So we have crossed here. These are the homozygous dominant genes for violet coloration, and these two are the recessive, the other parent who is having the recessive trait, that is white color. Okay, the white color. So now what are the individuals? The individuals that are showing the offsprings, probable offsprings, will all be heterozygous. This can happen in case 1. If suppose the individual, this unknown individual, is bearing homozygous dominant genes. If it is bearing heterozygous dominant gene, then what will be the case? The case will be this. The offsprings will be 50% heterozygous dominant and 50% homozygous recessive. Okay, so the individuals will be this. Now of course this we are working out. Okay, in practically when we are carrying it out, definitely this plant is 1, we cross it with a recessive individual and we will get some results, either this or this. So depending upon the results, we will come to the conclusion whether it was a heterozygous or it was a homozygous. Like say, if this is the case, then the unknown flower is homozygous dominant. It can be assumed because for the offsprings to be showing the dominant trait, definitely one of the genes, one of the factors, one of the alleles should come from each parent. Okay, contribution of the alleles 50% from one parent, 50% from the other parent. So you can see here, so for an individual to be expressing the dominant trait, at least one allele should be dominant. Okay, and so here you can see all the 4 possible offsprings are having a dominant allele. From where? Definitely they have got from the one of the parent who is, both of them are dominant alleles, possessing dominant alleles. The other case, okay, in the other case, as you can see here, so contribution of only 50% is of dominant allele, 50% is recessive allele. Now the recessive allele with another recessive allele will definitely give us a homozygous recessive condition, that is white in the particular example that we have selected. So this particular unknown violet colored bearing plant, this plant is heterozygous dominant. Okay, so this is how we carry out test crosses, okay, to find out the genotype of an unknown individual. Okay, now we will move on to chromosomal theory of inheritance. Now what is this chromosomal theory of inheritance? After Mendel put forward his theories of factors existing in pairs, some other scientists worked on it, and then they put forward a theory which we commonly call as chromosomal theory of inheritance. In 1902, Walter Sutton and Theodore Boveri put forward this theory. They said that genes are located on chromosomes, summing up the entire thing. Okay, so what did they state? They stated since gametes provide the connection between 2 generations, hereditary characters must be present in them. Because one generation, the parents, okay, suppose a particular generation is existing, the individuals are there, during reproduction, what happens in them? Meiosis takes place, gametes are produced. Gametes fuse, that results into the zygote. So now what is the link between the next generation and the previous generation? The link is the gametes. Okay, the link is the gametes. So gametes provide the connection between 2 generations. So through whatever is passing on from the parent to the next generation, through what is it passing on? It is passing on through the gametes. So they said that since gametes are the only link, so definitely the characters or the traits, whatever we talk about, must be present in the gametes only. That is only the source for the new generation. Now, where are these hereditary characters present? Now, what are these gametes? Gametes are cells. Basically, they are cells with a nucleus. Now, they say that hereditary factors are carried in the nucleus because it was, how did they confirm this? Because it was known by that time that the fusion of gametes to produce a zygote, what fuses? Not the entire cell. Only the nucleus fuses together. Only the nucleus, okay? Only the nucleus fuses together. The content inside the nucleus fuses together, not the complete cells. So it was confirmed that not in the other parts of the cell, but within the nucleus, the hereditary factors must be present. Now, what is present inside the nucleus? It was also known that inside the nucleus, chromosomes are there. Okay? Chromosomes are present. So what is fusing? If the nucleus fuses, means what is fusing? The chromosomes are fusing together of the 2 gametes coming from the 2 different parents. So, and later also it was found that chromosomes also exist in pairs. Already it was found out. Parallelly, things go on in with regard to scientific discoveries. So chromosomes also exist in pairs. It was also known. And Mendel had already expressed that alleles exist in pairs. That is, the factors exist in pairs. Now, what do the gametes have? Gametes have one set of chromosomes. Chromosomes are found in pairs within an individual. But when it comes to gametes, gametes have only one of the two chromosomes, one set of the two chromosomes, which again fuse together to re-establish the diploid state. That we already know. Gametes have 50% chromosomes. Okay, then two gametes each carrying 50% will combine together to restore that 100% state. That means haploid and haploid will combine together to re-establish the diploid state. The genes are located on the chromosomes. Okay, now what did Mendel state? Mendel stated that the law of segregation, what did he state? That the factors segregate or separate during gamete formation, and again they combined together in the F1 generation or whichever generation we talk about. They combine, they again get together. Okay, so again, what with regard to chromosomes, we can say the same thing. Chromosomes also get restored or come together during reproduction process when fertilization occurs. So both of them were combined together. Where will these factors, genes or alleles be located then? If chromosomes are combining together, factors are present in inside the nucleus, and inside the nucleus what is combining? Chromosomes. So definitely genes must be located on the chromosomes only. So during reunion of the chromosomes, genes are also getting reunited. So as there are two chromosomes of each kind in diploid cell, there must be two genes of each kind, one in each chromosome of a pair. So in a diploid cell, you have two chromosomes of each kind, homologous chromosome. What do we call as homologous chromosome? They are present. So, and what was stated already, the genes are located on chromosomes. So if there are two chromosomes which are again combining in the new individual, so each of the chromosome also must be having two genes, okay, of the similar type. When these two chromosomes combine together, the two genes of each kind also combine together to form a pair. So that is what is chromosomal theory of inheritance. That is, genes are located on the chromosomes, and when chromosomes reunite during the formation of a cell, of a zygote, the genes are also reunited. Okay, so that is the chromosomal theory of inheritance as stated by Walter Sutton and Theodore Boveri. So I hope you could get this concept. Okay, so if any further any queries or any questions, you can definitely put up in the comments section. Okay, thank you. Now, next is concept of dominance. Now, what is this concept of dominance? We will continue further here. What is the concept of dominance? Now, the concept of dominance, that is, why are some alleles dominant and some alleles recessive? This question must be very often coming to your mind. And why is it that some of the alleles, they behave as being dominant, suppressing the expression of the others, and why are others not expressing themselves in presence of an alternate trait? What is this? Now, to explain this, we have to go pointwise. Genes contain information for a particular trait. They contain the information, they contain the codes for a particular trait. Like say the trait is, the character is height, the trait we are talking about is tall. Okay, tall. The trait tall. Now, the gene responsible for making a plant tall will be having some information, some codes will be present in it whereby it will express to make the plant tall. Okay, so genes contain information. Now, in a population means many individuals of similar same type which can interbreed, that is a population. There will be many individuals, numerous individuals belonging to the same species, same type, okay, in a big group. In a big group, you may call it human or you may call it plant, okay, say a particular plant, say rose plants, okay, so many rose plants. So that all the rose plants taken together in an area, that will make the population of rose plants. So that way there may be present many forms of a gene. Like say you see different colors, okay, of flowers. You see different colors of flowers. Red you see, pink you see, yellow you see, of colors. Now all are colors. All are colors. Okay, that means all are genes. But definitely there will be some variation in the genes. That is why some flowers are red, some flowers are white, some flowers are yellow, or some flowers are pink color. So there are many forms of a gene. One form giving one color, second form giving a second color, third form giving a third color, or for any other character. Then a diploid organism has two copies of the same gene. Okay, this point you have to understand very clearly. That is a diploid organism. Okay, a diploid organism will have two copies of the same gene. That means, or alleles, two alleles. Okay, that is like say if I talk of the flower color of rose plant, red color. Okay, red color, it is due to the work of a gene. Fine. So in the plant, upon the chromosomes, there will be genes. Okay, there will be information for making the plant bear red color flower. Now that information, that gene, what we call as gene, it will not just be one copy. It will be two copies. Why two copies? Because there are two chromosomes. So on the two chromosomes, there is pair of factors, what we called, or what Mendel had called, pair of genes. So there will definitely be two copies of the same gene. Now same gene when I say here, the same gene when I say, that means the gene for color. Now the pair of the same gene may be exactly same, both of them, or they may be different. But whatever be it, they will be expressing for flower color. Here, these two genes may be same or different in the organism. Like say the example with which you are very familiar, I'll talk about that only. Like say height, plant height in pea plants. So plant height that is tall, it may be due to a homozygous condition. Okay, capital T, what we write as these are the two alleles or two genes. Okay, or different like capital T, small t. Okay, so they may be same homozygous or they may be heterozygous. Now let us try to understand it with an example. Okay, with an example, and an example which is also cited in your textbook. A gene E, let us take for example a gene E, capital E, for producing an enzyme that transforms a substrate S. Okay, that means a gene is produced in an organism. Okay, an enzyme is produced in an organism that will help in changing a substrate, anything. Okay, it will help in converting a substrate inside the body of the organism. That substrate we are naming as S. Okay, and the enzyme is formed due to the work of a gene which we are naming as E. Where is the gene located? Inside the nucleus. So the gene will express a protein, will help in formation of a protein, that is the enzyme, and that enzyme will help in transforming the substrate S. This is an example that we have taken. Let us see further. Now that gene E will have different forms. That means different alleles. Normal allele, okay, E, which is the normal allele E, it will produce the normal enzyme, which will clearly transform the substrate, change the substrate S to different forms. Now, during the course of evolution, when any organism is formed, it is said that they have the normal allele. Okay, so this is the normal allele E. This allele in the process, okay, throughout over the years, over time, what happens? It gets modified. The allele, the gene gets modified. Okay, and when it's getting modified, it doesn't mean the earlier one is not existing. It will continue to exist, but some of them will get modified in some individuals. It gets modified to produce E1, E2 and E0. Okay, three types. E1, E2, E0. So, how many forms of the gene E are present now? E, E1, E2, E0. Okay, so E may exist, may not exist. Okay, so here what happens here? E1, E2, E0 are the three alleles for the gene E. Okay. Now E1, it is seen that it is responsible, the gene E1 is responsible for producing the enzyme, which may be quite normal or maybe a little bit less efficient in transforming the substrate S. E2, okay, E2, the gene E2 may be responsible for producing the enzyme which is non-functional, which cannot walk, okay, which cannot walk. That is how the enzyme was produced. And E0, okay, E0, that when it got transformed to the form, the third form, which we are naming as E0, it is not at all capable of producing the enzyme. So the three alleles, okay, the three alleles, how they are getting modified, or how they are working. One type is either normal or less efficient, other type is non-functional, and the third type, it cannot produce the enzyme at all. Now, an individual may have any two combinations of this form, okay, because, okay, here you see a diploid organism has two copies of the same gene. So an individual will have two combinations of these forms, E1, E2 or E0. Any two combinations. Now these combinations may be homozygous, okay, as I've written here, it may be homozygous or it may be heterozygous. Okay, so here it might be EE, it may be E1 E1, it might be E2 E2, or it might be E0 E0, or it might be E1 E2 E0, or it might be E1 E2 E1 E0. I hope you got it, how the combinations can be formed. Okay, it will depend upon what the gametes are providing, from the what the gametes of the parents are providing, okay, to form the new offspring. Now, if the combination is E1, okay, if it is E1, phenotype remains unchanged. Why? Okay, phenotype remains unchanged means, in this case, okay, because what is it producing? The enzyme to transform the substrate S. E1, what is it producing? Normal enzyme. Okay, so even E1, both of them are producing the normal enzyme. So what happens? Ultimately, the expression in the individual will be formation of the normal enzyme to change the substrate S. If the combination is E2, okay, E2, what is E2? E2 here is non-functional enzyme. So here E2 will not express anything. Okay, it's non-functional. Even if it produces the enzyme, that means the enzyme cannot walk. Okay, it cannot walk. It cannot function the way it is supposed to. But it has an allele E. E can produce the normal enzyme. Okay, so upon whom, in such type of combinations, upon whom the ultimate expression will depend? It will depend upon the unmodified allele. Similar case of E0. E0 also no enzyme production. So it will depend upon E. So phenotype will depend on the unmodified allele E. Okay. So this presence of E2 or E0 along with E does not make any difference. It is almost like insignificant, but it has to be present, but its presence is insignificant as it is not producing any enzyme. So from this, what do we make out? Like say I'm just referring, okay, for this one, I'm just referring back. That is, in case of like say T small t, capital T small t. Okay, what happens? Tallness is seen, dwarfness is not seen. So presence or absence of small t becomes irrelevant. Likewise here, presence or absence of E2 or E1, E0 becomes irrelevant. Why? Because ultimate expression depends upon capital E, single E, which is producing the normal enzyme. So what do we see here? E and E1 are dominant. They will show the expression. E2, E0 can never help in expressing the enzyme. So they are termed as recessive because they are not being able to form that particular enzyme required in the individual. So that is, we get to know from this how the genes can be dominant or recessive. Okay, that's all for today. We will continue next day with a new concept on the same chapter. Thank you for your patient listening. Thank you very much.